So the big idea of this presentation, DNA Storage Opportunity and Challenge, is going to be the presentation of DNA being explored as a possible next generation storage medium. So normally when you think of DNA, you think of biology and genetics, but DNA is also fundamentally an information carrying molecule. So instead of storing genetic instructions, we are going to encode digital information into sequences of DNA bases. So this slide shows the basic workflow of DNA data storage. The process starts with ordinary digital information like binary data. That data is encoded into DNA sequences, meaning that the zeros and ones are mapped into combinations of the 4 DNA bases, being adenine, thymine, cytosine, and guanine. And then after encoding, the DNA has to be physically synthesized. This is considered the right step, where the actual DNA strands are chemically produced. Once those strands exist, they can be preserved in a physical container or solution, and then later when the DNA needs to be accessed, the DNA is sequenced, meaning that the order of bases is read back. Finally, that sequence is decoded back into digital information. The important point here is that DNA storage is a multi-stage system: encoding, synthesis, preservation, sequencing, and decoding. Because synthesis and sequencing involve molecular biology rather than pure electronics, latency is much higher than in silicon devices. That is why DNA is not ideal for fast access storage, but it can be attractive for archival storage where density and lifetime matter more than speed. To understand why DNA can store information, we need to go over its structure. So DNA is made up of nucleotides, and with each nucleotide contains one of 4 bases, A, T, C, or G. These bases pair in a predictable way. A pairs with T, and C pairs with G. That base pairing rule allows DNA to form a double-stranded helix. This is useful for storage because the sequence of bases acts like a code. Just as a hard drive or a flash memory stores information using physical states, DNA stores information through the ordering of chemical bases. So in this slide also goes over hybridization, which is when complementary DNA strands bind together. They do not always need to be perfectly complementary, but more complete matching generally makes the structure more stable. This matters because stability and reliable pairing are part of what makes DNA useful for long-term information preservation and later retrieval.
It might still be unclear to all of you how DNA can store electronic information, so we wanted to give an analogy to concepts we have discussed in class. The 4 DNA nucleotides can be likened to a multi-level cell flash memory, in which each base is assigned 2 bits of information. In this example, 1,1 is encoded to adenine, 0,1 to thymine, 0,0 to cytosine, and 1,0 to guanine. Different researchers have developed different encoding algorithms, so the values we have provided are simply illustrative and not a rule, but hopefully it helps paint the picture as we explain DNA synthesis. DNA synthesis is the process by which strands of DNA can be manufactured to an exact specification. This means that a precise chain of nucleotides can be arranged in any order to represent the electronic input data. This process is done nucleotide by nucleotide and can be very time-consuming, on the order of hours to days for a single strand. This currently makes synthesis the main latency bottleneck, and the reason why DNA is not a practical memory solution for any real-time computation. For this reason, DNA memory is being explored as an archival storage option for data not regularly accessed or modified. Unfortunately, synthesis errors and erasures do exist, meaning that you cannot reliably store all the data of a single file within one long strand without difficulty in reading it back. Thus, one option is to create smaller strands with primer targets and indices to help in error correction and data management. DNA is unlike computer memory in that addressing can't be done by using physical location on the chip. The DNA is within a solution, and location is not fixed. So the payload contains the actual electronic data encoded from binary, and the index identifies the addressing information.
Random access is an important advantage DNA storage systems have over traditional archival mediums like magnetic tape. This means that we can access the same amount of data from any location in about the same amount of time. From a high-level view, this is made possible by a system of keys and polymerase chain reactions. How this works is each DNA strand contains a set of keys at the beginning and end of the strand. Those keys work almost like file names on a file system. If you want to access the data on that strand, you must know the name of the file. So on an upstream system, these files are stored and encoded. So when you go to access it, you give it your file name or the key to the strand, and the polymerase chain reaction will flood the pool with the target that you desire. So when you go to read it, um, those strands take up most of the volume of the strands in the pool. Once the pool is flooded with the target strand from the PCR reaction, a sample can be taken to sequence or read the data. The most common method for this is sequencing by synthesis, where single DNA strands are read in parallel base by base using fluorescent markers. In the example, red pairs with green and yellow pairs with blue. Each time a marker is added, a picture is taken which captures an image of the markers. This marker is then chemically removed while leaving the base behind, where we then go to the next base in the strand. In the example, we are reading two single-stranded DNA samples in parallel. The first strand's first base is red, which pairs with the fluorescent green marker. The second strand's blue pairs with the fluorescent yellow. Once the picture is taken and those markers are removed, cycle 2 we can see that those first base pairs remain, while the fluorescent markers match on the second level, which is shown on that cycle 2 image. This process repeats until the strands are completely red. So the strands can be up to 5 bases, over 100. It just depends on how big the payload is. Another method that is being used is nanopore technology, where these DNA strands are forced through nanoscale pores, which generate a current depending on the base pair, and that can be used to read the data. Moore's law is a business observation made by Intel co-founder Gordon Moore, which states that the number of transistors on a microchip doubles approximately every 2 years. In scaling computing, this trend is desirable to keep up with the increasingly complex and large applications which demand more and more resources. The cost of sequencing DNA data was previously unfeasible compared to its digital counterpart. However, after following the trend of Moore's law for many years, recent innovations have accelerated past that benchmark, showing much promise for improvement and scalability in the DNA sequencing world.
So one of the big questions is why are we looking at DNA for storage and why are we so interested in it? So a key issue in the future is data storage landscape. Global data demand is increasing extremely quickly, and researchers have projected that by around 2040, global memory demand could reach roughly 3 times 10 to the 24 bits, or on a scale of yottabytes. This creates a materials problem. Conventional storage technologies, especially flash memory, depend heavily on silicon manufacturing. The concern is that projected silicon wafer supply may not be enough to meet future global storage demand if growth continues at this scale. So what this slide does is it frames DNA storage as a response to a scaling problem. If conventional materials become limiting, then alternative storage media needs to be considered. And DNA is interesting because it is extremely dense, so it can last a long time and requires very little power while stored. So this table compares DNA with hard disk, flash memory, and DRAM. The main takeaway is that DNA is not better in every category. In read and write latency, DNA is much slower than DRAM and not competitive with normal high-speed memory. That reinforces the idea that DNA is not meant for active computing memory. Where DNA becomes really impressive is in retention, power, and density. In the table, DNA retention is as greater than 100 years when compared with roughly 10 years for hard disks and flash memory. It also shows extremely low power requirements because stored DNA does not need constant electrical power to preserve information. The biggest advantage is volumetric density. DNA is listed at about 10 to the 19 bits per cubic centimeter, which is much higher than hard disk, flash, or DRAM. So the trade-off is pretty clear. DNA has poor latency, but excellent density, long-term stability, and very low storage power. So this slide is really meant to put the density advantage into perspective. So the quote on the slide says that if DNA level density could be achieved in information storage systems, all of today's global storage needs could fit in a 10 by 10 by 10 centimeter box. And they also estimate that around 1 kilogram of DNA could satisfy projected world storage needs in 2040. So the point is that we already have a commercial device doing this perfectly. Rather, this shows that the theoretical potential of DNA as a storage medium when compared with silicon-based storage. DNA can represent an enormous amount of information in a very small physical volume. So this is really why DNA storage is worth studying, because even if the technology is not yet mature, the density advantage is so large that it motivates the continued research in this field.
We hope now that it is clear to all of you the vast capabilities of DNA as a storage medium. The main takeaway from this presentation is that DNA is ideal for archival storage and has the potential for incredible breakthroughs in computing. The information density is enormous and far exceeds the capabilities currently achieved by magnetic tape storage. It has a much longer shelf life than magnetic tape, and DNA is also immune to many cybersecurity concerns that afflict silicon-based memory. Its biological nature means it cannot be attacked by digital hacking, and the way it is encapsulated safeguards it from most environmental attacks or accidents. It also has near zero energy consumption, making it the most energy efficient and sustainable data storage method. As for computation, the physical capability of DNA to pair with other strands, whether fully or partially, means you can perform massively parallel operations on the entire dataset. Polymerase chain reaction can replicate data at high speeds and allow for DNA to be used similar to hardware accelerators. The field of DNA research has come a long way since the double helix structure was first discovered in 1953. And thanks to next-generation sequencers, the research has accelerated even more since 2008. In 2012, the first in vitro DNA storage encoded 650 kilobytes of data, while in 2019, the University of Washington with Microsoft was able to store and successfully read back over 400 megabytes of data, which even included file formats like HD video and text documents in over 100 languages. The first commercially available DNA storage device was released in 2023 from the company BioMemory. And most recently, as of December 2025, Atlas Data Storage announced their Atlas Eon 100, which claims it will eventually be capable of 60 petabytes in 60 cubic inches, or roughly 1 liter. We wanted to make known some of the companies that are involved in DNA storage, should you wish to learn more. This first page is merely a snapshot of some industry leaders like BioMemory and Atlas Data Storage. This page also mentions some interesting projects happening, like the Library of Congress's official request for a DNA storage pilot program for their digital archives, and Southern University of Science and Technology working towards a different method of reading back stored DNA data in the form of a DNA cassette tape. There exist many more companies involved in this area of research, including a DNA Data Storage Alliance that seeks to employ international standards and ethics for the betterment and furthering of this field.
DNA storage has only become a possibility recently due to technology advances in various areas, which leaves many open questions remaining as the concept is still in its infancy. The trends for 2026 have split researchers across different areas. For example, here, computing experts have been publishing techniques to try and achieve full lossless storage. This is important for legacy applications, as any data corruption can be catastrophic. However, for AI-related applications, noise and loss can be tolerated to a degree. But to make an all-purpose storage system that is functional, losslessness must be achieved. Some of the techniques involve combining machine learning techniques with multiple independent reads of the data so that each base is read with a high degree of confidence. Error correcting codes and other classical computing techniques have also been explored to achieve losslessness. Another interesting development by the computing world is a DNA SSD by Mizzou, which is able to reuse the DNA in a finite space to achieve rewritability. The SSD uses a compact electronic nanoscale pore sensor for sequencing, which can enable its small size with a USB drive size device being the goal. On the other hand, biology researchers have been looking into avoiding costly deep freezing techniques, which is a barrier to archival storage on the computing side for storing data long term. Ensilocation is one technique that would add overhead to both the read and the write portion of the storage system, at the benefit of being able to store that data long term at room temperature with similar quality to the freezer. Because of this, we believe that tiered storage systems will be the first functional systems that could pioneer data storage for data centers. The data in this tiered system would be stored depending on when the next expected access would be. One of the papers published this year cut the synthesis time by half for ensilocation, so this tiered system may not be needed if the overhead eventually becomes negligible. We used these sources to back up our key paper in this presentation, and thank you for listening.